So welcome to video one in our course G08 on of metagenomics with respect to viral genomes here at the Purbright Institute. Please note that this course is a short course corresponding to one ECTS or two CATS credits. So let's briefly introduce the course. In this video, I'll talk about the resources and aims of this course, as well as an introduction on microbiomes and metagenomes. So here is a list of resources that you can use to extend your knowledge. The Galaxy training resources and carpentries for metagenomics are very, very good and very, very useful. There's also links to key resources and tools that we'll use, like Kraken2, CheckV, DREP, DeepVir Finder, VirSorter 2, Logan, and the Big Fantastic Viral Database as well, all of which can help you with your analysis, quickly focused on viral genomes. So what are we trying to do here? We're trying to assemble viral metagenomes, classify them, and analyze them functionally to a small degree in the time we have available. So really we want to look about viral metagenomics data and think about viral genome assembly, gene discovery, taxonomic classification, and content extraction, annotation, and interpretation as well. So by the end of the course, what we want to be able to do is consider different viral metagenome assembly approaches, apply taxonomic classification approaches to viral metagenomes, and compare your context with other published data. So please note that this course is not about metagenomics at a general level, nor about bacterial metagenomics, which is often the most common type of metagenomics. It's very much focused on viruses. So we're going to focus more on what viral metagenomics means. So if you look at the broad outline of metagenomics, we know why it's important in terms of examining livestock, maintaining livestock health, for animal welfare, and also to understand the evolution and spread of viruses in different environments and how that affects animal health and human health. So we can talk about microbes and the microbiomes as all the microbes in an environment that includes various particles as well as bacterial cells. In this way, we can also talk about the virome as all the virus particles in an environmental niche. And thus the virome does not discuss the viral DNA as such. Therefore, we can talk about metagenomes or viral genome, which is the DNA associated with the different microbes, of course, including viral RNAs and if it's an RNA virus. And of course, if you think of examples of metagenomes, it's pretty easy to think about where they are in us, on us, and around us, and why they're worth studying for different reasons. And please note that we're focused on viromics is not really the term that is useful for this course. We're more looking at metagenomes. So, what is metagenomes? It's the analysis of viral metagenomes sampled from an environment. So metagenomes can mean a full genome, a bit of a genome, a very, very mixed up jumble of genomes. It depends what you're looking at. So the analysis scales are very, very broad, and comparing between different studies is challenging because the data sets are quite different often. So what we want to do is look at viral genome and community structure and function as is relevant for our purpose. And we want to accelerate our understanding of genetic diversity in these viral metagenomes. And what we can think of this is that really we're kind of taking some sort of broad, untargeted sequencing approach that hopefully will accelerate our experimental approaches to discovering the diversity that's present, and also kind of avoid biases that can be induced by laborious steps in terms of culturing, passaging, and other things. So what we can think about is typical ways that we understand metagenomes. That often we have assays like PCR serological tests. We have this is different viruses that we study, and we can look at their genome structure. And one consideration is with all these tests that they're really good, but you only find what you're looking for. And you have to know what you're looking for in advance. So for well-known viral specimens or well-known types of certain viral organisms, our profiling may be limited to those that we know about a lot about already. and therefore metagenomics can help us discover more. So an obvious example is that we have a mammal of some kind that is infected with something that appears to be unusual. So what we can think about is that our steps include sampling, extracting the DNA, RNA, and conversion to cDNA, amplifying that using suitable technology, sequencing it, and then our main focus is on the bioinformatic interpretation of that data. So what you can see is that the full steps along the way all feed into each other, and therefore, if we're trying to do computational analyses, we need to understand the prior test steps to really understand what bioinformatic approach is most appropriate. If we don't have that sort of context in the data, then we're just wasting our time. It's not going to be useful. So we can think about different things. We can be looking at clinical samples or environmental samples, and to a large extent, the basic analysis steps are very, very similar. Often we'll be dealing with samples where there'll be some sort of post-cell component and a viral component, where the viral fractionation viral component will be extracted, and there'll be viral DNA or RNA that's extracted and sequenced. And then we want to do some computational analyses on that to interpret the genome or origin, taxonomy, the annotation, distribution abundance in that specimen, and links to the host, and other analysis that are relevant to us. So an example of this is the UKHSA are moving towards metagenomics approaches for a lot of, at least at a trial of metagenomic approaches for NHS work to accelerate pathogen analyses from a genome perspective, and in a less biased but also more rapid manner. So the key things here is think about outbreaks and how to identify the source of these outbreaks, and also to inform treatment approaches, whether you're looking at antibiotic resistance and the use of antiviral treatments, and consider the genetic basis of potential drug resistance in the pathogens of interest. So metagenomic approaches can support all of this. What we have to distinguish between viral metagenomics and bacterial metagenomics is that there's a lot of difference. So we know that viral genomes are quite diverse, whereas typically for bacteria, you're looking at a standard double-stranded larger genome. They tend to be structured, well-defined, whereas many viruses can be less so. We have many bacterial marker genes, whereas bacteria in viruses, we really don't. We can, we have knowledge of existing circular fragments or linear fragments for the different bacterial genomes, whereas viral genomes that can be a bit for a segmented virus that can vary a bit. The taxonomy is more clearly defined for bacteria in contrast to viruses because of their faster evolutionary rate. And here we can know that bacterial genomes often you get a whole contact, whereas with viral genomes, I mean, it's quite variable. The outcomes are very varied. And we know that the classification approaches, sometimes they overlap, like we'll use Kraken-2, but there are particular tools also that are useful for viruses to develop particularly for viruses. So we can really see that approaches for bacterial metagenomics and viral genomics are different. So let's think about different viruses. So is this the Baltimore scheme giving a sense of the DNA double-stranded single-stranded, RNA double-stranded single-stranded, segmented, and so on, retroviruses as well, and the different genome structures that these different viruses have, and how that therefore might impact viral metagenome approaches from the lab-based approaches to computational ones. So there's quite a lot. And we know that, sure, there's RDRP that is conserved to a good degree across many RNA viruses, the extent to which it will capture and differentiate between different ones is somewhat variable as well. So using a marker gene approach for viral metagenomics is going to be challenging unless we know we have a very defined data set in question to address. So we can think about, are we looking just at the whole metagenome, where we're not looking at a very targeted approach, we're just looking at whatever is there. We will get more information about abundance and other non-pathogenic viruses that are potentially present. Viral enrichment approaches such as using virus-like particles can be good. You tend to get higher depth and coverage. They're easier to assemble. You exclude diverse viruses. There is an issue with contamination for VLPs. Um, so there's kind of pros and cons to each approach. We also know that there's key steps that are shared across these things, and typically we're asking simply what or who is there, what organism, what virus is there, and functionally what may be the functions of the genes in terms of what is it doing. So what we can do is do things like de novo assemble the virus reads that we're able to extract them or map the reads. We can use something like Kraken2 to do taxonomic classification. We can search functional databases that are relevant to our virus. And we can also look at the relative rates of abundances there, which conceptually is similar to RNA-seq as well, that we're not just thinking about the sequences that are present, but the relative level of the different sequences too, which is the same as looking at transcriptional abundance in RNA-seq as well. So that brings us through these steps for an introduction to viral metagenomics. Please see the next video on metagenome sequencing.